I'd like to briefly define a few genetic terms. I think it would clear a lot of things up for patients. So, a proto-oncogene is a normal gene involved in cell growth and reproduction. They control the life cycle and allow the cell to replicate. But sometimes spontaneous mutations or environmental toxins like smoking introduce slight errors into the proto-oncogene. And in the setting of a normal, healthy immune system, the affected cell may actually self-destruct as a protective mechanism to, they'll repair the gene mutation, or an immunocell will notice that the cell has changed and will target it for destruction so that it doesn't, develop into full-blown cancer. Most mutations that occur in proto-oncogenes are quickly and efficiently remedied. In rare cases, though, sometimes the proto-oncogene mutation is allowed to persist. Our immune system misses it. And then another mutation may occur in another gene within the cell, for example, in like a tumor suppressor gene. So slowly, one by one, these minuscule mutations cause the cell to eventually become precancerous. There are very few genes in the body where, just one mutation in one gene causes this full-blown cancer. It's very much like a domino effect as far as developing cancer goes. And then there's, again, numerous stages within, a precancerous lesion, right? So the culprit gene at this point is then termed an oncogene, right? Because it carries the potential to shift the cell into a cancerous state. And the fail-safe switch has been deactivated. And so the cell can replicate indefinitely. Now, kind of a biology review here. DNA is translated into proteins, and proteins are what run the entire body. An oncogene is translated into an oncoprotein, okay? So today, many oncology medications target oncoproteins and mutated tumor suppressor proteins. So, for example, the oncogene HER2, which codes for growth factor receptors, expressed on the outside of breast cancer cells, that's a very common chemotherapy drug target. And so, when mutated, the number of receptors for this is markedly upregulated on the outside of the cell.